We just finished tallying up our energy from glycolysis, the gateway reaction, and the Krebs cycle. 36 ATP net profit. But we never talked about how do we take the energy stored in the carrier molecules NADH and FADH2 and turn that into ATP. That's what oxidative phosphorylation is all about. Oxidative phosphorylation is going to take place in the mitochondria. So I've drawn a picture of a mitochondria up here. We have an outer mitochondrial membrane and an inner mitochondrial membrane. I'm going to take a piece, a slice through the mitochondria, and I'm going to enlarge it right here. And everything that occurs here is going to occur everywhere else on the mitochondria, over and over and over again, probably several thousand times or more. This is going to be the outer mitochondrial membrane. This is going to be the intermembrane space. And then this right here is going to be our inner mitochondrial membrane. Outside of the outer mitochondrial membrane is going to be our cytoplasm of our cell. And within the inner mitochondrial membrane is going to be our mitochondrial matrix. So let's go ahead and start there. Most of the action is going to occur in the inner mitochondrial membrane. Here we have proteins. These are going to be called enzyme complexes. I like to call them enzyme complex 1, enzyme complex 2, and enzyme complex 3. I call them enzyme complexes because not only are they transport proteins, but they are also enzymes. Our last protein is a very special protein. This is going to be called ATP synthase. So let's go ahead and write that down, ATP synthase. It ends in ACE, so we definitely know it's an enzyme. And this is the enzyme / transport protein that's going to make ATP for us. So let's go ahead and start over here. Now our carrier molecules, our trucks, our loaded trucks, are going to come in and they're going to deposit their cargo here and here. So NADH carries the most high energy, so it's going to deposit its cargo at the first enzyme complex. So NADH comes in here, unloads its cargo, so it becomes oxidized to NAD+. The cargo, if you remember, was 2 high energy electrons, so there they go, 2 high energy electrons, and they're going to travel in pairs. And we also have our proton. There's our proton, it's going to get dumped into the mitochondrial matrix. These 2 high energy electrons are going to move through enzyme complex 1. And as they do, enzyme complex 1 is going to siphon off just a little bit of energy. And it's going to use that energy to pump protons from the mitochondrial matrix into the intermembrane space. Now every time a pair of electrons moves through, we're going to pump approximately 1 proton. These 2 high energy electrons now need to move to the next enzyme complex, but it's too far away, and these enzyme complexes really don't move. So we need another little shuttle molecule, and that little shuttle molecule is going to be called Q, or ubiquinone. And ubiquinone is capable of moving back and forth. It's going to come over, it's going to pick up these 2 high energy electrons and transport them over to enzyme complex 2. Enzyme complex 2 is then going to siphon off a little bit of their energy to take a proton from the mitochondrial matrix and actively transport it or pump it into the intermembrane space. From here, our electrons are now going to move to enzyme complex 3. But same problem, it's too far away. We can't just throw the electrons. So we need to have another shuttle molecule. This one is going to be called cytochrome C, and I'm going to write CYT, cyt C, cytochrome C. And it's going to be attached to the surface of the inner mitochondrial membrane. So these 2 high energy electrons now move to cytochrome C, and cytochrome C brings them over to enzyme complex 3. Enzyme complex 3 then uses a little bit of their energy to take a proton and pump it or actively transport it into the intermembrane space. From here, these 2 electrons now have very little energy left. Let's call them low energy electrons. And they're going to move down here, and we have to get rid of these low energy electrons. If we don't, the electrons are going to begin to back up, back up, back up, back up, and eventually all this is going to shut down. We don't want that to happen. So what's going to be waiting for these electrons right here, and this enzyme complex is going to catalyze this reaction, is going to be oxygen. So I'm going to write 1/2 O2. I'm going to write 1/2 O2 because oxygen in the air looks like that. And oxygen double bonded to another oxygen atom. We're going to break this chemical bond and produce two separate oxygen atoms. The way we write that is 1/2 O2. So we've got one oxygen atom here essentially. These two low energy electrons, there they are now going to get added to the oxygen. So oxygen is going to get reduced. We're also going to take two protons that are floating around in the mitochondrial matrix here and add them to oxygen. Ultimately, we're going to produce a water molecule. And then the water molecule will leave this area and become part of the other water molecules in the cell. But the gist of the story here is we have removed the electrons, and additional electrons can now move through here without backing up. So oxygen, because it performs a very important process, is going to be called our final electron acceptor. Our final electron acceptor. And then oxygen, when it receives electrons and two protons, ultimately will yield water. But oxygen is going to be our final electron acceptor. Now let's talk about FADH2. FADH2 carries two high energy electrons and two protons, but it can't dump its cargo off here because its electrons just don't contain as much energy. So it's going to have to go to the next enzyme complex, which is enzyme complex 2 right here. So FADH2 comes in, unloads its cargo, and is oxidized to FAD. Now when it unloads its cargo, the two high energy electrons move through enzyme complex 2. As they do, we're going to pump another proton because we're going to siphon off a little bit of the energy. And the two protons that it was carrying get dumped into the mitochondrial matrix. These two electrons are then going to move to cytochrome C and then ultimately to enzyme complex 3, which is going to siphon off a little bit more energy and pump a proton. So the process repeats itself. The FAD and the NAD+, because they're carrier molecules, they now go back to the previous cycles that we talked about, glycolysis, the gateway reaction, and Krebs cycle to pick up another load of cargo. We're not going to waste them. We're just going to recycle them. They're going to pick up energy, unload energy, pick up energy, unload energy, over and over and over. Let's go a little bit further here. We're constantly pumping protons from the mitochondrial matrix into the intermembrane space. Because of this, we ultimately are going to acquire a high proton concentration in the intermembrane space. And because we're constantly taking them out of the mitochondrial matrix, we're going to have a low hydrogen ion concentration in the mitochondrial matrix. Because of this, we have a concentration gradient. Think of this high hydrogen ion concentration is like water stored behind a dam. We have lots and lots of potential energy stored here. What's going to happen is these hydrogen ions are going to look for a way out. And the only way out is to move down their concentration gradient through ATP synthase. And as these hydrogen ions move through the ATP synthase, that's where we're actually going to make ATP. Now, because we got a flowing river of hydrogen ions here, we're going to call it a proton motive force. A proton motive force. Think of it as like a river of flowing water molecules, except that it's a river of flowing hydrogen ions. Now, if we want to get energy from a flowing river, we'd stick a water wheel into it. And as the water turns the water wheel, we then generate electricity. Well, it's the same thing here, but instead of having water, we're going to have hydrogen ions. And as those hydrogen ions flow through here, we're going to tap some of their energy of motion and use that energy to make ATP. We're literally going to take some of that energy of that flowing hydrogen ions and store it in the chemical bond between the last two phosphate groups on ATP. So the end result here is to make ATP. So an ADP plus an inorganic phosphate, which are floating around here, are going to get connected together, and the energy that creates that chemical bond comes from this flowing river of hydrogen ions, the proton motive force. So we're going to make ATP. And this is what we want to make right there. Lots of ATP. All this ATP is going to be made from the cargo of FAD and NAD+. Now let's go back and look at this a little bit more. These three enzyme complexes are going to be referred to as the electron transport chain. So let's write that down. I'm going to call it the ETC, the electron transport chain, because it is a chain of transport proteins that move electrons. Over here, this is going to be referred to as chemiosmosis. Now, osmosis is going to be the movement of water molecules through a semi-permeable membrane. Here, this is chemiosmosis, the chemi referring to hydrogen ions. This is going to be the movement of hydrogen ions through a semi-permeable membrane. And that is going to be it for cellular respiration.